Today, we are going to learn about bacteria, one of the earliest living organisms on Earth. Bacteria are organisms that can survive in any type of environment, whether it is very cold temperatures or very hot temperatures. They can survive in any type of environment. In this lecture, we will cover the following topics: the definition of bacteria, the structure of bacteria, including its components, the classification of bacteria based on different characteristics, how bacteria reproduce, the differences between useful and harmful bacteria, along with examples. So let's start. Do you know that when this world began, at the time of the world's beginning, the first living organism was bacteria? Bacteria are organisms that can survive in any type of environment on Earth, whether it is very cold temperatures or very hot temperatures. They can survive in any type of environment. If we talk about bacteria, bacteria are unicellular organisms belonging to the kingdom Prokaryote. There are a total of 5 kingdoms in the world, and in the kingdom Prokaryote, those living organisms exist that do not have a true nucleus or cellular organelles. Therefore, bacteria also do not have a true nucleus or cellular organelles, which is why we have placed bacteria in the prokaryotic kingdom. Now we will read about the structure of bacteria. This is the structure of bacteria in front of you. In this, you can see that the outer boundary of bacteria is called the cell wall. The function of this cell wall is to protect the bacteria from the external environment. Then you can see this circular DNA, which is inside the bacteria and is called plasmid. The other DNA is chromosomal DNA, which is the bacteria's own DNA. It means that bacteria have two DNAs. One is the plasmid, which we call circular DNA, and the other is chromosomal DNA, which is the bacteria's own DNA. This fluid-like material present inside the bacteria is called cytoplasm. Then you can see this thread-like structure, which we call flagellum. The function of flagellum is to help the bacteria in movement, to move from one place to another. It uses the flagellum. Now we come to the classification of bacteria. The classification of bacteria is done based on the different features and characteristics of bacteria, such as on the basis of shape. After that, we will study the classification of bacteria based on the composition of the cell wall. Then we will study its classification on the basis of the mode of respiration. After that, the final classification we will study of bacteria is on the basis of the mode of nutrition. So these are the 4 classifications of bacteria. Let's come to the first classification on the basis of shape. On the basis of shape, bacteria are classified into 4 categories. The first is cocci, the second is bacillus, the third is spirilla, and the fourth is vibrio. In cocci, the bacteria are of spherical shape, circular, round. These are called cocci. Bacteria that are rod-shaped are called bacillus. Bacteria that are spiral-shaped are called spirilla. The final classification is the fourth, which is called vibrio. These are the bacteria that are comma-shaped and they are called vibrio. If we talk about examples, an example of Coccus streptococcus pneumonia, which causes pneumonia. The second is bacillus, an example of which is Escherichia coli, also known as E. coli. The third category is spirilla, an example of which is Spirillum volutans. The final category of classification is vibrio. An example of vibrio is Vibrio cholerae, which causes cholera disease. Now let's come to the classification of bacteria on the basis of cell wall composition. The cell wall is composed of different substances, and based on this, bacteria are classified into two categories. The first category is called peptidoglycan cell wall. Bacteria that have a cell wall made of peptidoglycan are called peptidoglycan cell wall bacteria. Bacteria with cell walls made of Lipopolis asaccharides are called Lipopolis asaccharide cell wall bacteria. Now let's discuss examples of both these categories. Gram-positive bacteria come under peptidoglycan cell wall. Gram-negative bacteria come under lipopolysaccharide cell wall. Now let's come to the classification of bacteria on the basis of the mode of nutrition. Nutrition means how they arrange their food. This classification is divided into two categories. One is autotrophic bacteria and the other is heterotrophic bacteria. Autotrophic bacteria are bacteria that can arrange their own food, meaning in the presence of sunlight or chemicals. Using chemicals or sunlight, they produce their own food, so they are called autotrophic bacteria. The second type is heterotrophic bacteria, which cannot produce their own food and depend on others, so they are called heterotrophic bacteria. If we talk about examples, cyanobacteria are examples of autotrophic bacteria. Most disease-causing bacteria are heterotrophic as they depend on others and infect others. These are the two categories of classification of bacteria based on the mode of nutrition. Now let's come to the classification of bacteria on the basis of the mode of respiration. Bacteria are divided into two categories based on the mode of respiration. One is anaerobic bacteria and the other is aerobic bacteria. Anaerobic bacteria. The word anaerobic is made up of two parts. One is in meaning no or never. Here I will explain something. Whenever a word has an a or an attached to it, it means no or never. So here we have anaerobic and meaning no and aerobic meaning requires air. These are the bacteria that do not require oxygen for respiration. So they are called anaerobic bacteria. And bacteria that require oxygen for respiration are called aerobic bacteria. If we talk about examples, an example of anaerobic bacteria is actinomyces. Now let's discuss an example of aerobic bacteria, such as Mycobacterium, which comes under aerobic bacteria. Now let's discuss the difference between useful bacteria and harmful bacteria. I will tell you that not all bacteria are harmful. Some bacteria are beneficial to us. I will provide examples and explain what benefits we can get from bacteria. The first benefit we get from bacteria is that certain bacteria convert milk into curd. These are useful bacteria called lactic acid bacteria. The second type of bacteria helps ferment food, which we use for fermentation, and such bacteria are called streptococcus and bacillus bacteria, which help in the fermentation of products. The third type of useful bacteria helps with digestion and improves the immune system of our body. These bacteria are called actinobacteria. The fourth type of useful bacteria helps in the production of antibiotics, which we use for the treatment of bacteria infections, and such bacteria are called soil bacteria. Now let's come to harmful bacteria. These are the bacteria that cause different types of diseases in humans. These diseases are called infectious diseases. For example, I will mention pneumonia, tuberculosis, diphtheria, syphilis, and tooth decay. These are diseases caused by bacteria, and the treatment for these is through antibiotics only. But if anyone wants to prevent these bacteria from causing diseases, then various methods are used, which we call precautionary methods. For example, sterilization or disinfecting of exposed surfaces or instruments and tools. Everything we use, we sterilize or disinfect it so that the bacteria present on it are destroyed. When they are destroyed, they die, and the bacteria present on those instruments or tools will not be able to cause disease. Other methods include the application of heat, disinfectants, ultraviolet radiation, pasteurization, and boiling. These methods are also used for disinfecting bacteria or sterilization. Question of the day. What term describes bacteria that require oxygen? Only a genius can answer in the comments below. If you would like to watch my other lectures on topics like the lungs, kidneys, liver, brain, and eyes, visit my channel to view my other videos.